Good morning. Microalgae has a wide bioactive composition, which are usually rare or absent in other taxonomies. The derived substances have diverse secondary metabolite uses. Cosmeceuticals are oral or topical cosmetics with drug-like benefits. Exhibiting economic potential, they are utilized commercially. Estée Lauder's Pure Purifying Cleansing Jelly, La Mer's Age Prevention Superfood Mask and Daniel Juvence's Beauty Mask Peel. Skin whitening is a multi-billion-dollar industry with invasive procedures like bleaching, chemical peels, steroid cocktails and intravenous injections. Harmful methods warrant the development of safer skin whitening products. UV radiation stimulates free radicals, thus activating the tyrosinase enzyme. This induces melanogenesis, creating melanin in pigment. Macroalgae extracts are utilized to achieve hypopigmentation, able to suppress COX-2 expression and proliferation in mouse models. Firstly, a unialga must be isolated in a sterile environment. Mass culturing then takes place before processing into a cosmeceutical product. But, what's missing? How do we extract the intended bioactive compound from the micro? The first method of extraction is the supercritical carbon dioxide and ethanol co-solvent of carotenoids from Nanochloropsis gaydetana, Syncococcus sp and Dunliella salina. Carotenoids are natural lipid-soluble pigments in phytoplankton, marine algae, plants and some bacterial species. They inhibit tyrosinase activity. Some examples of carotenoids that inhibit tyrosinase activity are crocusatan H, crocin and. Extraction begins by loading extraction cartridges with homogenized raw materials. These are left for 15 minutes. A micrometric valve regulates the flow of 4.5 millimoles per minute at 508 degrees C. Products are collected in glass tubes containing ethanol. Solvent is removed via nitrogen stream at 408 degrees Celsius in dark. Ideal conditions are 5% ethanol, methanol and dimethylformamide. Optimum pressures and temperatures in gaydetana and D salina are 400 to 500 bar, 600 degrees Celsius, Syncococcus sp. are 300 to 400 bar, 518 C. Astaxanthin is a powerful antioxidant with tyrosinase inhibition. It can be manufactured synthetically or derived from Haematococcus pluvialis. H. pluvialis is a freshwater microalgae containing a high amount of Highest astaxanthin concentration occurs at aplanospore development at vegetative cell growth stage. Scientists can deconstruct the cell wall and induce production. Induction is conducted via centrifugation and rinsing in NIESN medium. Cells are then transferred to fresh medium and incubated in growth chamber at 25 degrees Celsius. Cells are suspended in ionic liquids. 1 milliliter culture aliquot is collected after 12 hours germination and washed with distilled water. Astaxanthin is extracted from the cell pellets by vortexing at 28 degrees C. Solution is centrifuged again before remaining cell paste is treated with half a milliliter IL and incubated for 24 hours in darkness. Liquid extraction was conducted with 1 milliliter ethyl acetate. Optimal condition was determined as pre-treatment with 1-ethyl-3-methyl-imidazolium methyl sulfate for 1 minute and 12 to 18 hours germinate. Fluorotannin 7-fluoroethyl and diethyl of Eclonia cavo inhibit tyrosinase activity in the bottom epidermis. Successful reduction of zebrafish pigmentation has been observed. By quantifying the total fluorotannin content measured per the Folin-Sioccult methodology, it was determined that the optimal conditions were 100% ethanol solvent for 32 hours at 50 degrees Celsius with pH 7. Supercritical CO2 and ethanol co-solvent is fast, environmentally friendly and produces high yield, but inducing astaxanthin production and extraction has high yield in a controlled environment but may not be applicable to other.
Conventional solvent liquid extraction is cheap but damages the environment. Generating high yield of astaxanthin is imperative to meet commercial